Hi, everybody. Thank you for that introduction. Absolute pleasure to be here talking about my favorite topic, which is corals and coral reef and wider cnidarians. So, yeah, I'm the marine invertebrate curator at the WA Museum and also have a research group at Curtin University here. So, we're getting involved in lots of different things in lots of parts of the world, but I'll just tell you a little bit today about corals. So the plan for today is I'm going to introduce you to the phylum Cnidaria, of which the hard corals belong. We'll talk a little bit about taxonomy, especially in relation to the hard corals. Along the way of the discussion, I'll just talk a little bit here and there about some cool aspects of coral biology and ecology. And at the end, I'll just highlight the vulnerability of corals and what's going on in Western Australia and coral reefs of the world at the moment, because there's a lot. So today, let's just kick into learning a little bit about Cnidaria. So the Cnidaria are an ancient group. They date back at least 700 million years, and that's based on the fossil record. But as you'll find out, a lot of these Cnidaria are soft bodies, so they don't preserve well in the fossil record. So these Cnidarians could be even older, up to 1 billion years old. So there's at the moment estimated to be about 13,000 living species, but that is a vast underestimate of the diversity that's out there. For most marine invertebrates, we think we know about 1/4 of the diversity that's in the oceans. So there's likely to be at least 3 times more species than that. And we're still waiting to find those and also to formally describe them. So the cnidarians are living all over our oceans. They're mostly in the ocean, in the salt water, but there are a couple that live in fresh water. They've occupied habitats everywhere, from the shallowest coral reefs to the deepest ocean trenches. So they're in the tropics where they love the warm water, but they're also in the temperate waters and even in Antarctica. So the cnidaria have also all size ranges. You can get minute cnidaria like the irukandji, but doesn't mean they can't pack a punch, of course. And then also the massive corals like parietes, bommies that can be the size of a house. But one of the really special things about all the cnidaria, and the reason why they're called cnidaria, is because they have nematocysts, which are stinging cells. And the word cnidos is Latin for nettle. So it means nettle-like, stinging-like. So we'll talk a little bit more about the cnidaria and their nematocysts shortly. So when it comes to the phylogenetic tree of life, the cnidarians are very low in the tree. They're early evolving animals, and they're actually one of the animals that's the first to evolve a multicellular life where they've got true tissues. So they've got their tissues differentiated into different layers. So they have a 2-cell layers with an ectoderm and an endoderm. So the things that are older than the cnidarians are sponges. So the sponges do not have true tissues. There's no tissue organization. So the cnidaria are really important because it's the first time animals evolved tissue differentiation. And then as evolution continued, the animals just became more and more complex through time. So when we think about the cnidaria, there's a huge diversity, all types, all shapes, living everywhere. So we're trying to, through taxonomy, make sense of it. We're trying to understand this diversity and classify it and arrange it in a way that seems logical for the human brain. So amongst the cnidaria, what has emerged through years and years of research from many different people is that there are 3 subphyla. There's the Anthozoa, the Endocnidozoa, and the Medusozoa. So today we're just going to talk about the Anthozoa and the Medusozoa because the Endocnidozoa are just a very small, unique group of microscopic parasites. So we won't spend too much time on those. Within the Anthozoa, are the hexacorals, and this is the subphylum within the corals exist. The corals are hexacorals, but then there's also the octocorals, which is otherwise thought of as soft corals. So the hexacorals don't just include the hard corals, they also include anemones and corallimorphs and zoanthids. And the octocorals also include all the sea fans, the Helioporidae, which are blue corals, and the Pinatulacea, which are sea pens, which you can see on the top right in that image. So all of these anthozoans are sedentary adults, and they have motile larvae, planktonic larvae, but no medusal stage. So as opposed to that, we have all the medusozoa. So these are all the box jellyfish, which are called cubozoans, the hydrozoa, which are hydroids and things like the blue bottles. So that image, the second image there with the sand, that's a giant benthic hydroid that was found in the Ningaloo canyons off the West Australian coast, probably at around 2,000 meters deep. So it's standing about a meter high. So they're basically a carnivorous predatory hydroid. So very cool animal, that one. Then we get the Scyphozoa, which is all the true jellyfish. So you might have seen a lot of jellyfish in the Swan River. They're all the Scyphozoans. And then there's this other cool group called the Staurozoa, and they're a stalked jellyfish. So those ones actually only live around hydrothermal vents, and I've never seen one of those, and I'd really love to. So just trying to click through here. The thing about listening to that, you can see there's a whole range of shapes and sizes and sedentary lifestyles versus pelagic lifestyles. There's no end of variety. So it's hard to kind of imagine, like, how are all these different animals actually related to each other, and why are they all, grouped within this phylum together? So in another way of saying that, you would say they're not immediately recognizable as being monophyletic. And monophyletic means descending from a common ancestor. But actually, there's a lot of really important reasons as to why they are monophyletic. So one of them is that they have radial symmetry. So this is where you can divide a coral colony or a jellyfish or an anemone in various planes, and they will be reflective of the opposite plane. So kind of like dividing a pizza up, cutting up a pizza into equal shares. It would look similar on the opposite side. So that's radial symmetry, and something that all the adult cnidarians have. But just remember that as a larvae, they actually have bilateral symmetry, which is more similar to what the beetle there has. A second thing that all these cnidarians have is either a medusal or a polyp life stage. So all the jellyfish have the medusa, which is the floating body form, but the corals and other animals have a polyp. So that's their juvenile attached phase. So for the medusae, the mouth is on the underside, and in a polyp, the mouth is on the upper side. So having either a medusae or a polyp is something that all the cnidarians do. Something else that we mentioned very briefly earlier is the cnidocyte. So this is a really important trait of all the cnidarians. So the cnidocyte, otherwise called as a nematocyst, is basically just a stinging cell. So it's a really highly specialized cell. It's very, very cool. It has basically this little barb that's inside a single cell, and it's wound up, and it has a little cilia, a little hair sticking out of the cell. And when something brushes up against that hair, like a fish or a little piece of zooplankton, it'll trigger that barb to fire. And then there's venom inside that barb. So once one nematocyst triggers, it often triggers all the ones around it as well. And this is, how these corals are preying on food. So some of these nematocysts are actually sticky. Some anemones have a sticky cells. So they're using a slightly different mechanism, but the same concept of predation. So very, very cool specialization of these cells. The last thing that is a shared trait between all the Cnidaria is this double tissue layer that they have. With the ectoderm, it's kind of like the outside skin, and the endoderm that surrounds the gut. And then in between, there's this layer of non-living tissue that's called the mesoglia. So let me just move on to the next one because my slides are jumping a little for me, but hopefully they're stable for you. The other cool thing that's really interesting about all these Cnidaria, and particularly the corals, is that they are absolute feeding machines. So they have those nematocysts so they can actively catch food in the daytime and or at nighttime for some, and that's called heterotrophy. So they're actually using predation and catching their own food. So they're catching zooplankton and small things like this. On the top right image there, you can see a coral that's partially ingesting a shrimp. So this is something I observed in Dampier. So that is a very large animal for this coral to be ingesting, and I've never noticed anything like that before. We're not sure if it was caught it while it was alive or whether it actually had died and then the coral has somehow happened upon it and it is trying to ingest it. But that just shows you the size of animal that this particular coral is trying to ingest. So it does have all these enzymes in its mesenteries inside its gut that can break down all the nutrients that it absorbs through things that it digests. And then those nutrients pass through those different cell layers. The other thing that corals and some of the other photosymbiotic cnidarians do is they photosynthesize. So just like plants that have chlorophyll, corals and other animals within the cnidaria can have these little cells, which are shown in the circle here, which are called zooxanthellae. So those zooxanthellae are symbiotic algae, and they are photosynthesizing. They live inside the tissue in a symbiotic relationship. So they're getting a home from the coral's tissue, and they're living in there, and they're photosynthesizing and getting energy from the sun. So when they do that, the byproduct of that absorption of sunlight and photosynthetic pathway creates sugars. So the sugars are the byproduct that the coral tissue is living on. So the coral's on a sugar high all day long. It's getting all these sugary byproducts from photosynthesis. And then at nighttime, it puts its tentacles out and it's feeding, it's catching prey overnight. So basically the corals, these other cnidarians, they're feeding 24 hours a day. So they're absolute feeding machines and absolute weapons at, deriving sustenance. Something else that's pretty cool about all these cnidarians is they don't have a normal nervous system like humans. So just remember they're one of the most early diverging animal with the true tissues, the true cell double layers of tissue. But they do have a web of nerves. So they have nerves. It's just a different kind of development of a nerve network. So I mentioned when one cnidocyte fires off, the neighboring ones will also fire, but also when the corals have their tentacles out, if you touch a coral tentacle on one side of the colony, it will respond and all the tentacles across the entire colony will retract. So there is communication going on within a colony and between these different animals. So remembering every one of those little circular polyps there with the tentacles in the ring, that's an individual animal that lives in a colony with all its neighbors. So they are communicating with each other. So it's a very basic nerve net. So that's quite fascinating, which has obviously developed into the central nervous system in all the higher animals like us. So corals also have the ability to detect light, and not just corals, all the cnidarians have these very simple early forms of eye spots and genes that are triggered by light. So cubozoans, things like the irukandji box jellyfish, they have these things called ocelli, which are basically eye spots, and they really can detect light, especially between light and shadow. So occasionally if you're in the water and you've got maybe like a little southwest stinger and you can see it coming towards you, and it actively changes direction because it can sense that you're there, especially the shadow of you, and it's trying to avoid the shadows, and it can make directional changes and respond to things. So while it doesn't have eyes like us, it has these capacity to detect light. And this is developed in corals in their reproductive biology because they are detecting moonlight, and that's one of the cues for mass coral spawning. So the corals have been found to have genes that are very primitive genes that have developed into eyes in higher animals. So they have this PAX-C, which is thought to be a photoreceptor gene. And as I say, one of the sort of early genes that's even in humans that has just further developed as, animal diversity has developed in higher forms. So cnidarians are also really important for the habitat. They are the engineers of all of these marine ecosystems, especially on a coral reef, but also in all the other types of ecosystems in the ocean. There's a lot of animals that live in the soft sediment between coral reefs, and often these are called filter feeding habitats or sponge gardens, and all the soft corals and anemones and sea pens and all these other cnidarians live in that kind of habitat. So the coral reefs are really important, but there's also all this other stuff going on, and the cnidarians are forming that habitat. And once there's habitat, that attracts other animals that live on that habitat, that they feed on that habitat, gaining protection from the habitat. So here's a few examples. We have a little crinoid on the top left on that yellow sea fan. That's a type of echinoderm. The middle picture at the top is an eel that I saw out in the Pacific Ocean living in this big colony of Goniopora, doing a beautiful job of camouflaging with it. On the right-hand side are some mollusks living on another sea fan. There's plenty of fish that live in amongst the Acropora corals, and all those little blue chromis are always absolutely stunning to see on a reef. And then here we have the little pom-pom crab that has anemones stuck to his legs. So these are just some examples of the incredible variety of associations that occur between all the diversity of life in the marine environment and their habitat. But all the cnidarians are also really important for their interaction with humans. And I'll just give you a few examples here. Of course, we know about the negative interactions when people get stung by things like a box jelly. But on the bottom left, for example, is a little statue made out of a coral that someone brought to me at the museum to try and find some information about. And I went on a deep dive, and from what I could work out, that this is a little statue from the Torres Strait. And they often carve these statues out of corals, and they put them in the garden to look after their crops when they go out to sea fishing. So it's a very totemic protector object, so culturally important. They're using corals actively to have this role of protection. And then in the top middle there, you can see what looks like some rocks, but actually these are a whole heap of corals that have been formed into these tombs. And this is in a part of Micronesia in the North Pacific Ocean, a location I went to, and I realized that these tombs where they used to bury their kings in this ancient culture called Leia, they built their tombs out of corals. And when I investigated more about this story, about these tombs and the corals, there's ancient folklore talking about the people handing the corals from hand to hand across the reef. They actually harvested live corals for building purposes to bury their special people, their kings, in these tombs. So that's another very spiritual link between corals and people. And then when you go down the middle, you can see this young chap here with a really excellent smile. He is chewing betel nut. So betel nut is another cultural practice around some Pacific islands, and these guys are in Papua New Guinea. They actually harvest coral, live coral, and cook it on the fire, and that becomes the limestone that they chew with a bit of lime leaf and the nut of the palm tree. They chew the lime and the coral, and that's their sort of cultural practice that they're exercising out there in the Pacific. On the top, obviously, there's tourism and the curio industries, which has been utilizing corals for a very long time. And at the bottom, there's the aquarium trade, which is actually a huge industry, and lots of anemones and corallomorphs and different types of hard and soft corals are traded around the world, just tons and tons every year. But in the middle there, these little tubes is another very interesting thing about cnidarians, because a lot of them have a lot of pigments, and you can extract those pigments and make natural products out of them, and also the other chemicals in these cnidarians and other marine invertebrates like sponges are packed full of chemicals. And some of those chemicals have therapeutic purposes. They make anti-cancer drugs or anti-inflammatory drugs out of these natural products derived from invertebrates such as cnidarians. But something really important about the corals is they're often a bright red color, and that bright red pigment has been extracted from corals and is used in biomedical research, and particularly cancer research, because that fluorescent red color is inserted into a cancerous cell, and then they can track the movement of those cancerous cells inside the body. So there's so many applications and interactions between all the cnidarians and humans as well. And as we just wrap up this little section on cnidaria, I thought I'd show you, if it's going to work. Yeah, there it goes. This cool spot at the Abrolhos Islands. So if anyone knows the Abrolhos, they're just off the coast of Geraldton. And this is the patch of anemones. So this is highly unusual. Anemones usually occur just as individuals, but this is a whole garden bed full of anemones. And as you can see, there's anemone fish all through there. And they've made this huge aggregation. So it's a really, really cool thing. And I'm not really aware of anywhere else in the world where this giant patches of anemones has been encountered like this. So all the adults are there mixed in with young ones, and they're all busy defending their territories. And yeah, it's quite a scene and quite a spectacle. So just recapping on that, these cnidaria, they're really cool. They're really ancient. They're simple animals, but they have the earliest, organizational things and the earliest development of eye spots and nervous systems that has led to development of those things in higher animals. They have those nematocysts, which is the key thing that all the cnidarians share, and also this symbiosis that they have with zooxanthellae is another reason that they're really, really successful. So now let's move on to a little bit about hard corals. And so that's all the scleractinian corals and their taxonomy. So first, I just want to talk a tiny bit about taxonomy. So taxonomy is how we're trying to classify animals in relation to who they're related to, who are their sister species, and who are they most, who's their most common ancestor is what we're trying to resolve. So again, just trying to make sense of all the diversity amongst corals. So in the original days when corals were first described, people were describing them and classifying them based on only morphological features. But as time has gone on, especially in the last few decades, the modern taxonomic approach is using molecular data as well. So elements about the genome. So we started with just looking at single gene regions, and now we're starting to look at whole genomes to try and resolve the relationships between individuals and then between species and then at higher levels. So when you're doing this genetic work, you're really just trying to sort these animals into who's related to who, and then they end up in a phylogenetic tree that's showing clades, and you want those clades to be differentiated in a way whereas everything that's within a clade together has the same common ancestor. Most of the time when you're looking with molecular data, you're finding more species and more diversity. So the more you look, the more you find, quite often is what we're having. Sometimes the molecular data matches morphological data and sometimes it doesn't. So it can be quite complicated to try and interpret the taxonomy of corals, and it also depends on what species concept you're using. So in the past, we've often used a biological species concept where you're saying that species don't reproduce with each other. But these days, people are more and more using genetic species complexes. So they're just talking about they have unique genetic signatures. So there's a lot of difference in how people interpret relationships, especially at a species level. So its work is ongoing and evolving as we go. So when I was trying to simplify coral taxonomy for my colleagues and my students, I made this coral identification guide for Western Australia, which is called the Coral Compactus. And you can find this on the West Australian Museum website and download it for free as a PDF. It's actually a few years old now, and I'll tell you a couple of changes that have happened since then. But in general, there's 16 families recognized today, families of coral, and about 80 genera in Western Australia. And within that, there's probably about 450 species of coral in Western Australia. So this guide is only getting you to the genus level because it would be a lot of work to get a full guide full of all the species level IDs for WA. I'd love to do it someday, but we're not there yet. So since I made this, which was a few years ago, there's been quite a lot of changes in coral taxonomy. When I made it, various genera were called incertae and that means we don't know where they belong. So you can see them on the bottom right of this figure here. Since I made that ID guide, people have gone ahead and described new families to accommodate those genera. So we now have these brand new families called family Leptestriidae, Plesiostriidae, Plerogaridae, and Ulastriidae. So those corals have now found a home. So they have their own family. So they're very unique, evolutionarily distinct from everything else. And then within the existing families, there's often multiple genera. And in orange there, you can see family Lobophilidae. A big change also happened only a few years ago, which is one genera called Symphilia was absorbed into another genera called Lobophilia. So at the moment, there are no Symphilias. All the species that previously were belonging to the genus Symphilia are all now called Lobophilias. So there's these changes that are ongoing and basically means that I need to update this identification guide. So when you start doing coral taxonomy or taxonomy for any group, whether it be birds or plants, there's always a lot of terminology. And when you're trying to, read a species description and understand why one species is different from the next, you really need to have a good grasp of all that terminology. So I'm just going to introduce you to some of that now, because you would need to kind of familiarize yourself with some of these terms. So first, I'll just talk about the polyp, which is the tissue component of the polyp. So the polyp is a single individual living animal that is one of hundreds of thousands in an entire coral colony. And that polyp is the one that lays the corallite. So it's the skeletal structure, the little tube that's around each individual polyp. They have the tentacles, which are so important for not only feeding, but they also have zooxanthellae in the tentacles. And then there's the coelenteron, which is the gastric cavity directly below the mouth. And that's where all the action happens, because that's where the mesenteries are. And the mesenteries are really important for digestion and respiration. And that's where all the gametes, the egg and sperm bundles, will line up on the mesenteries prior to coral spawning. So there's a lot happening in the gastrovascular cavity inside those mesenteries. They're really pulling their weight, those mesenteries. I'm just going to show you a couple of just random little things on the side here. This bright orange coral is called a sun coral. This one is photographed at the Abrolhos Islands. It's actually beautiful. So if you look below, that other little orange thing is a nudibranch. And this nudibranch lives in the coral and eats that particular coral and ingests its pigments. So ingests that yellow color so it can camouflage in on that particular coral. So it's only associated with this coral, and it has embodied that coral so much through, utilizing all that orange colored pigment that it's actually genetically identical now. So that's quite fascinating. If you were trying to identify that coral through that nudibranch through a molecular approach, it would come back as matching the coral. So that's really quite unusual. And then on the bottom is another very fancy nudibranch. So obviously nudibranchs are mollusks that have no shell. But this one is really cool because there's a few nudibranchs that do something special called kleptonidae, and that's where they eat and steal nematocysts from corals and other types of cnidarians. And they don't digest the nematocysts. They actually just store them in their rhinophores, which are their like tentacle looking things off their body. And then they'll use them for defense. So they can steal and store the nematocysts, which is a really cool little adaptation that these nudibranchs have evolved to survive out there on the reef. So going back to some more of the terminology, this is all about the skeleton. So when you're identifying the corals, especially in the old school approach, which is a large component of what I do, you're looking at the coral skeleton. So you want to remove the tissue, and you usually just use household bleach to remove the tissue. And then you're looking at all different aspects of the coral skeleton. You can be looking at the corallite itself, and you can be looking at the calyx. So you're measuring things like the size of the calyx. The callus is like the upper edge of that corallite, the width of it, how thick the callus wall is. The synostium is the skeletal material between the corallites. You'll be looking at that and how much synostium is there. Is it bumpy, or is it smooth, or is it porous? Is it blistered? There's all these different things that happen to the synostium. The septa are really important because the septa are those projections that are going into the center of the corallite, and that's where the tentacles anchor onto the septa. So that's the anchor point for the tentacles. Now those septa can have a whole variety of shapes, sizes. They can have teeth on them. They can be in different cycles. So you're counting how many septa there are and looking at the size of them and the shape of them. And then you come over to the right-hand side there. There's a word called the columella, and that's in the very center of the corallite. You can have different shapes. You can have like little pillars or a little mesh of skeletal material forming a columella. The piliform lobes form at the end of the septa near the mouth. So those are little upright projections that come on the end of the septa, and when they're really well developed, and every septa has them, they form what's called a piliform crown. So these are all the things you're looking at. You're looking at the size of the corallites, the shape of the corallites, whether the septa, how many cycles of septa you've got, the dentation on the septa, whether they've got teeth or not, the columella, the shape of the columella, whether they've got piliform lobes. You're looking at all these tiny, tiny details of the skeleton when you're trying to identify things at a species level. These things change. But when you first trying to identify a coral, you really start off looking just at their overall morphology. And this is something you can obviously see in the field. You're looking at, are they like massive form? Are they branching form? Are they encrusting? And throughout this taxonomic journey, it's really just a process of elimination. You're trying to eliminate everything that it's not. And then it just leaves you with a few options of what it is. So for me, you see on the second image on the left, there's a mushroom coral. You see something that shape, you know straight away it's one of the corals within the family Fungidae. So you can just mentally eliminate everything else because nothing else has that shape. And then on the bottom right, you see one called meandroid with all the tentacles sticking out. Again, there's only a couple of genera that have that shape. So straight away, you've eliminated heaps and heaps of things. So all the different shapes are useful in taxonomy. So once you know what morphology you've got, in many instances, it narrows it down already to you for just a number of different genera. And for example, within the family Merulinidae, which is a big family we'll talk a little bit more about in a minute, the next thing you would do to try and resolve which genus you've got is you'd be looking at the shape of the corallites. So are they spheroid? And spheroid means they share a wall with their neighbor. They touch each other. Or are they plocoid, which means they have their own wall. They're separate to each other and there's synostium between the corallites. So once you knew that, it tells you if it's spheroid, you're into Favides or Silastria or Leptastria type of corals. If it's plocoid, you've probably got a Dipsastria, a Cyphastria, or a Plesiastria. So all the way you're just eliminating and trying to narrow down your options. When you encounter something that's meandroid, and some of you might have heard that as a brain coral, there's only a few genera have that growth form. So the Platygyras, Leptoria, Lobophilias. So straight away, you've sort of focused in on your most likely groups. So there's plenty of easy ways to quickly get yourself orientated to what genus that you've got. There's a few other cool shapes like this one called Phaceloid, and in the aquarium industry, they call these torch corals. They look a little bit like torches, like the Olympic torch. There's a few genera do that, the Lobophilia and the Corlastria. And then you have the Flabellomeandroid. So there's, meandering valleys with big gaps in between. So there's, again, only a few different genera have that shape. And then there's Phaseoflabellate. So this one at the bottom, it's often just a couple of corals have that, and they have these really fleshy mantles and very, very pretty when they're alive and underwater. So you can kind of see you're looking at gross morphology, then you're coming into the corallite shapes mainly. But on the top right, you can see what's called a scanning electron microscopy photo. And in some instances, all of this type of big information about colony shapes is not enough, and you need to get right into the detail of the nitty-gritty and look under an electron microscope to see really fine details of the skeleton to be able to resolve what species that you've got. So there's a long sort of list of things you need to look at, ending up at the most microscopic features for some things to be able to resolve between species. So what I'm going to do now is just talk about just a handful of the really common and important families out on the reef. So family Acroporidae is a very important one. And I'm just going to talk about the genus Acropora. These are often called branching corals or staghorn corals. And the word Acropora means acro is single and pora is pore. So a single pore, and what it's referring to is that axial corallite that you can see in the red circles. That's a living animal at the tip of every branch. So the axial corallite extends all the way down through the middle of the branch, and it's leading the vertical growth of a branch. On the side in the yellow circle are the radial corallites. So there's many more radials, and they are leading the lateral growth, the width of the branch. So the Acropora are the only corals that have an axial corallite, so a living animal at the tip of the branch. So as soon as you're looking at something, because there's a few corals that have a branching form, you need to look and see, is there an axial corallite, a living animal at the tip? And if there is, then straight away you've got an Acropora. So then it's just the big question of which Acropora have you got, because there's a very diverse group. And when you were trying to work out which Acropora have you got, you'd be looking at the shape of those radial corallites, because they can have all these different shapes. Some of them are a tube, some of them have tube with extended openings. Some of them are pressed up against the branch. And you can see in this coral that I've got a picture of here, this is one called Acropora millipora. It has all these very lipped radial corallites forming these really even extended lips. So that's a very distinctive shape and something that's only done in probably about maybe 3 or 4 species of Acropora have radial corallites like that. So as soon as you saw that, you narrowed all the options of 450 species down to about half a dozen at most. So these are really important on the reef. The Acropora, they grow really fast, but unfortunately they're really, really vulnerable. They're one of the ones that will be the first to bleach in a heat wave event. Storms will damage them. All the Drapella, the crown of thorns, all love to eat them. So they make really important 3D habitat for all the fishes and other animals to live in, but they are often in a boom bust kind of lifestyle, or they'll grow really fast and then die really hard, really quickly. So on the contrary to that, we have something called Parades, which is often one of the really long-lived species, especially when they form the big, massive colonies the size of a house that can be thousands of years old. And they're often quite resilient to things like heat stress. So it's more of a resilient coral. This one can form a whole range of growth forms. So you can have branching parietes as well as encrusting ones and massive ones, but all of them have this tiny corallites. So you can see in that picture there, each corallite is only about 1 millimeter wide. So there's only one other genre, and it's called a Montipora, and it's one of in the family Acroporidae that has such tiny corallites. So if you looked at a coral and you saw these tiny 1 millimeter corallites, you'd be straight away, it's either Parades or Montipora. You can eliminate all the others based on that. So then it becomes really, really tricky to work out what species you've got, because you need to get those skeletons under a microscope and look at all that detail about the septa and the cycles of septa and the paliform lobes and different features within that 1 millimeter corallite to try and work out what you've got. So it's quite a complex and difficult task for the Parades. Then below that, you've got the Goniopora. So this is just an interesting one I wanted to show because this is one of the few corals that has its tentacles out all the time. So all day and all night, it's got its tentacles extended. So it's preferentially heterotrophic feeding, so catching its food rather than photosynthesizing as much as other corals. So this is one of the corals where if you touch some tentacles on one side, it would retract across the entire coral colony. So yes, these gonioporas are pretty cool and pretty obvious once you see. Then there's the Paradidae. So this is another coral that's a fast grower, like the Acropora. It often comes in and takes over in a disturbed habitat and can, some people call these weedy type corals, but that's a bit of a harsh term. They're just successful early colonizers and early successional species, so they come in straight away. But that's really important because they provide some of that initial habitat for all the other animals. So the thing about Pocillopora that makes them obvious is they have all these bumps across their synostium and their skeleton, and those are called verrucae. So those extended bumps that are sticking out, and you can see there's corallites, the little circles all across the verrucae as well. So no other coral has these bumpy things that are called verrucae. So if you saw a coral and you see those bumps, you're like, right, that's verrucae. We've got a Pocillopora. So straight away you can eliminate all the other options and you're down to a Pocillopora. And again, from there, you're just trying to work out which species you've got. And then at the bottom, we've got Ceratopora, which is a relative of Pocillopora within this family. So Ceratopora is sometimes called a needle coral. It forms these fine branches. Is solid skeleton, unlike the Acropora. If you recall, it's got the axial corallite at the end. So, well, something's happened there. So this is solid skeleton at the tip, no animal at the tip. Those corallites are lined in rows, and it's often a beautiful pink color, as you can see on the bottom left there. On the right-hand side, you see that little cage-looking thing, and that's been formed by a little gall crab. And this is a really fascinating little association that occurs between this crab and this coral. So the female gall crab will sit on the branch of the coral and it will nip away at the coral as it's growing, and it will be manipulating its growth to grow into this little cage. And what that cage is its reproductive chamber. So the gall crab female is quite large. It'll be sitting inside there for its entire life. The male gall crab is very small and it can come in and out. But once the gall has grown and the female has manipulated the coral to grow into a full little cage, the female is stuck there for life. It's not leaving and it'll die in there. But that's its little brooding chamber. So that's a fascinating little relationship, and it's always surprising to me how often I see those out on the reef on this particular coral. So it's not a rare thing. You'll definitely see it if you're out there having a snorkel. All right, and then we're nearly at the end of going through these, but I just wanted to mention this family, Marylinidae, because it's so diverse. Like you can see that all the different genera that are within this family. So this family used to be called family Favidae. You may have heard of that name, but because of some advanced genetic work, we resolved that everything living in the Atlantic Ocean and the Caribbean was separate to everything that lives in the Indo-Pacific. So it led to a name change for all the Indo-Pacific Favidaes, and now they're all called Marylinids. So within this family, very diverse family, there's some very common corals. One is called the Dipsastrea, and as you can see, the Dipsastrea has the plocoid corallite form where the circular corallites have their own walls and they're separate to each other. So that they're also quite large. They're like at least a centimeter, sometimes 2 centimeters wide, every individual corallite. So as soon as you see plocoid corallites about a centimeter or larger, you know you've got a Dipsastrea. As opposed to that, below you've got favites. So favites have the steroid corallite form where they're more angular and they share walls. So there are a couple other genera have this steroid growth form. So you could have a Coelastria or a Favites or a Leptastria doing that. But you've still helped yourself eliminate some things when you see the shape of that corallite. And then we'll get into these ones, which is Cyphastria. So they have the plocoid growth form as well, but those are really small. They're probably about 1/2 centimeter across. So the Dipsastrias have the big plocoid correlates. Cyphastria has very small plocoid correlates. So very easy to differentiate from each other. And then you get this one, Echinopora, which is kind of in the middle. Those correlates might be just under 1 centimeter. But the thing about these is they have all those spines on the synostium. You can see them really well developed, making it look all fuzzy almost. So that's a distinguishing feature of the Echinopora. Even though the Cyphastria has a little bit of spines, it's nowhere near as fuzzy, and those correlates are much smaller. So you can kind of see how you can differentiate between these different genera. And then again, at the bottom of those brain corals, the Platygyra, very, very obvious because of their growth form. So I won't go further with all the different genera because, you know, we'll be here all day. It's like a lifetime of work because corals are really, really diverse. But just like the corals themselves on the reef are evolving, they continue to evolve. So does our understanding of the corals. So our taxonomy evolves, and that's why we have name changes and different information, different lines of evidence makes us revisit our understanding of evolutionary relationships and tweak it as we go. So we're slowly trying to crack this coral code, but it's quite a massive task, and we certainly have not cracked it yet, but it is an absolute work in progress. It can be pretty tricky to keep up with all the name changes that are going on and understand what's the latest, you know, understanding of the evolutionary relationships of one species to another. So fortunately, there's this website that you'll find very useful if you keep on the taxonomic journey in your future careers. It's called the World Register of Marine Species. And this is like the authority place where all the information is contained about what species names are accepted and what is their higher taxonomy. So what family are they in, what class, what phylum, all this information is summarized at this website. So whenever at the museum we're registering a new specimen, we will look at the worms website and align our classification and our taxonomy to what is in there to keep it standardized and keep it current between museums and around the world, different researchers trying to use our data and use our database to find things. So last of all, I just wanted to briefly touch on this one. So you've probably heard this year about the big marine heat wave that has affected Western Australia, and it's actually been the biggest heat wave event we've had because the entire West Australian coast, everywhere from Ningaloo Reef up through the Pilbara, around Broome, all through the Kimberley and out to the offshore atolls of Ashmore Reef, Scott Reef, Raleigh Shoals, they have all succumbed to coral bleaching this year. And that's the first time the entire Northwest Shelf has been so devastated. But it's actually just the tip of the iceberg because corals have been in trouble for a while. I was involved in this study back in 2008 where we did the first red list assessment of the threatened status of corals at a global scale. And back then, that's almost 20 years ago now, coming up to 20, we were resolved from that 1/3 of the coral species in the world were threatened with extinction back then. And I've just helped with a huge team of people to revisit these threatened species assessments. And now 50% of the corals in the world are threatened with extinction. So corals are really in trouble, and the problem for them is that there's this thing called cumulative impacts. They are being hit by one heat wave after another. On the Great Barrier Reef, we're up to like 7 heat waves in the last decade. So the corals need to be able to grow up to maturity. So they need to be 3 to 5 years old to reproduce. So even the surviving colonies reproduce. They provide little larvae that settle on the reef. Those larvae might make it up to a year old, but then the next bleaching event comes along and they die. So there's all these setbacks that the corals are constantly facing on the reef. They'll get a few steps forward and a few more back. So not so much in Australia, but in other parts of the world, corals are really threatened with pollution, and there's crown of thorns starfish outbreaks and dropelia outbreaks, and so many different threats to corals and to the survival of corals. But really the big one, the big leveler on a global scale, is climate change. And the corals are really, really susceptible, especially things like the acropora. So they really are in trouble. So we need to rapidly learn as much as we can about the corals, and taxonomy is a really useful part of that, because if we want to protect reefs and we want to protect their functioning, we need to know all the elements, the parts, the working parts of the reef. And it's really hard to protect something that you don't know. And how can we protect biodiversity if we don't even know what's out there? So I encourage you all to carry on your marine science journeys and hopefully get into marine science careers and university studies, and navigate yourself towards working on biodiversity and working on protecting all our incredible marine life and marine ecosystems, because there's plenty more to do.